Today we're going to talk about differences between DNA and RNA and the process of transcription, which is part of this protein synthesis that we're learning in class. It's the first part of protein synthesis, actually. So first of all, let's take a look at the differences between the two. DNA is double-stranded. RNA is single-stranded. DNA uses deoxyribose, that's where the D for DNA comes from, and RNA uses ribose, that's the R for the rRNA. Now again, if it ends with OSE, we're talking about a sugar. Deoxyribose and ribose. So this has a different sugar in its sugar phosphate backbone than the RNA here. This uses ribose. The phosphate's still there in here just as it is in there too, the phosphate groups. What's also similar is the fact that we have adenine, guanine, cytosine in RNA, just like we do in DNA. But what RNA doesn't have is thymine. Instead, it has uracil. So when RNA is being paired up with DNA to make the messenger RNA that will leave the nucleus and go to the cytoplasm, to the ribosome, to be read to build a protein. When it does that, anytime there is an A on the DNA strand, we'll see it paired up with a U on the RNA strand when we go through our process using RNA polymerase, the enzyme, to build this RNA molecule. Last time during DNA replication, we used helicase and DNA polymerase as our enzymes. So, double-stranded, different sugars, single-stranded, has uracil, has thymine. All right, let's take a look at the process of transcription here. So we're moving from DNA into RNA. So here's our enzyme. And just like helicase, RNA polymerase is going to split and help unwind our DNA molecule here. Now there are promoters, sections in DNA that tell the RNA polymerase where to bind to and where to unwind. But this is just going to give us an overview using this model here. So let's say the promoter was here and we start to unwind. As we see, it's very similar to our DNA replication model. Okay, so I have it unzipped a little bit here, and this is our active site. This is where messenger RNA is going to be built, and it will come off and go this direction here and then leave the nucleus and go to the cytoplasm. As this moves through, what we'll see is our DNA strand unzipped. It will come back together and be zipped right back up as it leaves. Now, each of these strands have a name now. This is our template strand, and this is our non-template strand. A template is something that you're going to make a copy off of. So we're going to make a copy off of this strand here. So this is our template strand. So here's our active site. And again, T will be paired with A. That doesn't change. I'm going to match up our shapes here. And A will be paired with U now, as we move through our active site here. And then what will occur here is, again, we'll have a split, because again, we want this strand here to leave. We don't want it to stay with our template strand. We want our template strand to come back with our non-template strand at the end. So let's keep on going here. C with G. As we move through, and then we have another A with U. Keep going. C with G. We have a T here, which, remember, RNA does have an A, so we can pair an A with that. We'll keep on moving. Keep on pushing through. And so what we're starting to see on the opposite end here now our template strand and our non-template strand are coming back together. And eventually this would continue to go on until eventually we get to a point where in the DNA strand it would code to stop. And then the coding would stop and the entire mRNA strand that has been built would then break off and go into the cytoplasm, where it would then be received by a ribosome to be read. This copy would be read, because remember these are instructions to build a protein. It will be read to bring in the proper amino acids. And the ribosome will read these 3 at a time, a codon at a time, and each codon will code for a particular amino acid. And they will be bonded together to make a protein. But that's not until we get to translation.